photosynthesis and Calvin cycle reactions occur in the bundle sheath cells. And basically what the plant is doing is it's figured out a way to pump CO2 into these bundle sheath cells and increase the concentration of CO2 to make it less likely that Rubisco will run into an oxygen and more likely that it will run into a CO2. And it much prefers to fix CO2 than oxygen, so it almost eliminates the photorespiration problem. And it almost eliminates oxygen binding with Rubisco and RUB. So how does it pump the CO2 in? That's where the second cell layer, this is the mesyl cell layer, is important. So CO2 is a gas. It'll come in and it'll dissolve into the aqueous part of the cell solution. And it gets taken up by another enzyme called PEP carboxylase. Okay? PEP carboxylase doesn't fix oxygen, it only fixes CO2. And it catalyzes the reaction between CO2 and PEP, which is phosphoenol pyruvate, to make oxaloacetic acid, or oxaloacetate, which is a 4-carbon compound. Okay? That 4-carbon compound gives C4 photosynthesis in its name. It's converted to malates, pumped across the cell membrane into the bundle sheath cell. It's decarboxylated, and you get pyruvate. Remember pyruvate from the end of the glycolysis. So you see a lot of the same compounds. It's sent back into the basophil cell. An ATP is required to get back to phosphoenol pyruvate, and that cycle continues. So basically, an extra cycle is added. This cycle's purpose is to get CO2 into the bundle sheath cell. The cell wall here is sort of impermeable, so CO2 can't leak out easily. And so it basically pumps up this concentration of CO2 in that bundle sheath cell to eliminate Rubisco binding with oxygen. All right. Questions? There's a cost to this, and that cost is 1 extra ATP to do this extra set of reactions. Remember in non-cyclic electron transport in the light reactions, how the paradoxin could go back and make more ATP? The cyclic electron transport, being able to make a little extra ATP is very useful for that step. So if you're a C4 plant, you're doing a little bit more of that cyclic electron transport because you need a little extra ATP. Great. Note also that there's this spatial separation, and that spatial separation is important. So carboxylase acts in the mesophil cell, Rubisco acts in the bundle sheath cell, so there's a spatial separation